Good morning, students. Myself, Major Komal Katyal, Department of Zoology, GK Govani Government College, Bhinmar. Today, we will discuss about my third lecture on immunology. In this part, we will discuss about the various cells involved in the immunology or parts of the immune system. So today's topic is cells of the immune system. The cells of the immune system consist of lymphocytes. They are the T lymphocytes, B lymphocytes, plasma cells, memory cells, the NK lymphocytes or the natural killer cells or the NK cells, then monocytes, macrophages. Granulocytes include neutrophils, eosinophils or acidophils, and basophils. These are the main cells of the immune system. We will discuss one by one. Now, the cells of the immune system are broadly divided into two lineages, one the lymphoid lineage and the other one is the myeloid lineage. The lymphoid lineage is basically responsible for giving rise to B lymphocytes and T lymphocytes, and apart from it, it is also responsible for giving rise to NK cell or the natural killer cell, as it is clearly visible from the diagram. The myeloid lineage is basically responsible for giving rise to remaining WBC and their modifications. It includes the granulocytes like neutrophils, eosinophils, and basophils, which later on forms the mast cell, the monocytes, which later on forms the macrophages. Now first we will discuss about the B lymphocytes. They are named as B lymphocytes because in mammals their development occurs in the bone marrow. In the case of birds, the development occurs in the bursa of Fabricius, and hence we call them the B lymphocytes. They can be divided into two subsets on the basis of response. The one response is T cell independent polysaccharide binding response, and the second one is T dependent protein antigen binding response. On the basis of 5 different types of immunoglobulins produced by them, they can be divided into 5 subtypes. The end cells of the B lymphocytes lineage are the plasma cells, although rarely seen in the blood plasma. Now, B lymphocytes, as we know, are responsible for the production of immunoglobulins or the antibodies. How they function is depicted in this slide through humoral immune response. First of all, the B lymphocyte recognize the specific antigens, and after recognizing the specific antigen, they proliferate and differentiate into antibody secreting plasma cells. These antibodies bind to specific antigen on the microbes and destroy microbes via specific mechanism or the humoral mechanism. Some B lymphocytes evolve into the resting state and they are known as the memory cells. These memory cells are responsible for keeping the information related with a specific antigen and what kind of antibody is required to deal with such kind of antigen. The next type of response is the cell-mediated immune response. The cell-mediated immune response is controlled by the T cells or the T lymphocytes. It is also depicted through a diagram in this slide. First of all, the T cell recognizes the peptide antigen on the macrophages in association with the MHC class or the major histocompatibility complex class. And after identification, it can also identify the molecules on the cell surfaces. It helps the body to distinguish self from non-self. Apart from it, the T cell goes into the effector cells stage and that is able to kill the infected cell. So basically they are also responsible for carrying out the phagocytic activity, as it is also visible with this diagram. The T lymphocytes are named as T lymphocyte because they are produced in the thymus, and there are two types of T lymphocyte as per the WHO classification. Here we will follow the classification in which we have classified T lymphocyte into four different types. The first type of T lymphocytes are known as helper T lymphocyte or CD4 plus cells. The CD4 plus T cells activate phagocytes to kill microbes. Apart from it, they also activate other T and B lymphocytes to perform their functions. The second type of T lymphocytes are the suppressor T lymphocyte or TS cells. The suppressor T lymphocytes are responsible for suppressing the action of the other types of T lymphocytes. The third one, which is the main type of T lymphocytes, are the cytolytic T lymphocyte or CD8 plus cells, or they are also known as cytotoxic or killer T cells. The CD8 plus T cells destroy infected cells containing microbes, and these infected cells are actually tagged by the antibodies or certain microbial proteins. And after their identification, the cytotoxic cells perform the process of phagocytosis. Some of these cytotoxic T cells can recognize and destroy cancer cells or the tumor cells. The fourth type is known as the delayed type of hypersensitivity T lymphocytes or TD cells. The TD cells cannot be easily differentiated from the TH or the helper T lymphocytes, but they are very commonly occurring in those tissues where delayed hypersensitivity reaction is occurring. Now, next about the NK cells, or which are popularly known as natural killer cells. The natural killer cells are the large granular lymphocytes or the LGL. They are neither B lymphocytes nor T lymphocytes. They play an important role in the innate immune system. As you know, innate immune system is the inborn immune system which is present in our body by birth. These natural killer cells kill virus or bacteria infected cells. It means the virus and bacteria, which are the intracellular pathogens. As we know, there are two kinds of pathogens. One are known as intracellular pathogens, which actually invade the cytoplasm of the healthy cell and make them infected cell. And the second type of microbes are known as intercellular pathogens, and these intercellular pathogens are basically present in the extracellular fluid or in intercellular spaces. They kill these virus and bacteria which are present in the infected cell without the aid of antibodies or the complement system. It means they don't require any type of antibody or the complement system. More or less, they are working like a pistol, and from this pistol a bullet is fired at the virus or bacteria infected cell. They represent the first line of defense against the tumor or the cancer cells, as also shown in this diagram. These natural killer cells contain certain azurophilic granules in their cytoplasm, and these azurophilic granules are basically known to be source of lytic factors or the perforins, and these perforins basically penetrate the infected cells and thereby killing the infected cells. Now, how these natural killer cells distinguish between an infected cell and a healthy cell is shown through this slide. As you can see, the mechanism of killing by NK cell. If you look at this slide, in the first one, a healthy cell is shown at the surrounding the healthy cell on the surface. The major histocompatibility complex class 1 is attached, and when NK cell recognizes these healthy cell, it never fires a bullet from its pistol. Now, in the second case, when an infected cell that does not present a MHC1, it means major histocompatibility complex class 1, and it is infected. In this slide or in this diagram, it is depicted through a pathogen virus. The infected cell is basically infected by a virus, and now this NK cell releases the granules with perforins and proteases. It means the bullet of NK cell consists of azurophilic granules or perforins and proteases, and these proteases are those enzymes which are responsible for killing the infected host cell as well as the infecting agents or the microbes. So by the presence or absence of major histocompatibility complex class 1, its presence or absence determines the NK cell to recognize whether a cell is infected or healthy, and if it finds an infected cell, it fires the bullet in the form of perforins and proteases, thereby killing the host cells as well as the microbes. So this finishes our first part on cells of the immune system. Thank you.